We calculated pseudotime, cluster-specific regulatory networks. We did some basic analysis of these regulatory networks and silico perturbation analysis, and we examined how the perturbation affected the trajectory. I'm going to do a quick tutorial covering this recent work done by Samantha Morris's lab. They created a suite of tools called CellOracle, which can do a variety of things, including gene regulatory network analysis, and importantly, this in silico gene perturbation. So instead of having to run an actual experiment where you knock out a gene in cells or an animal model, you can actually simulate a knockout using your single cell data. For this analysis, you only need single cell RNA data, but if you have ATAC data, you can construct these GRNs with your cell-specific data instead of having to use a more general GRN so that analyses might be more accurate. So they have published very in-depth documentation on GitHub. It goes more into the nuances, so I do recommend checking that out as well. So first thing you want to do is create a new conda environment. They recommend Python 3.8, so we're just going to stick with that. And then we'll activate that CellOracle environment. And then we're going to want to install several Python packages, including CellOracle. And don't worry, I'll put this on my notebook in my GitHub along with the code. So I just started a new Python notebook in the same miniconda environment. And we're going to start by loading in a bunch of modules. So I installed and I'm importing SEVelo here just because I'm going to use one of the datasets instead of using just the CellOracle tutorial dataset, just so there's some variability. So we're just going to load in the pancreas developmental dataset, which has 3,696 cells and 28,000 features. And just to make this data a little more raw-like, this is completely optional, but I'm just going to get rid of some of this data, like the neighbors and PCA, the UMAP coordinates, highly variable genes. I'm just going to get rid of that. I didn't really need to do that. I just wanted to clean it up a little bit. All right, so the first thing we want to do is save the raw counts, which is currently under X, in a new layer called counts. And we're going to filter out the genes that aren't expressed. We could do a pretty thorough pre-processing, and if this is your data, you should, but I'm using the assumption that these data were already pre-processed. So I'm just going to go straight to normalization instead of filtering out any cells. So I'm just going to put this disclaimer here, just in case people are following along but not watching the video. So I'm just doing basic normalization and doing log1p on my counts, and then I'm going to save these log1p counts in the raw slot of my A data, and then we'll calculate the highly variable genes. We'll do PCA neighbors, and then cluster with laden. I have a pretty low resolution here. Of course, you would change this depending on your data. And here's where there could be a lot of variation. Just to keep it simple, I'm just going to do basic UMAP, but you know, this is actually a pretty controversial subject in the field right now. Does 2D reduction actually represent the data correctly? Which reduction does the best job? A lot of people still use UMAP, of course. In the Morris lab analysis, they actually do a force reduction, basically where each cell is a node and they're connected by little springs based on their neighborhood graph. If you're interested in doing it that way, you can check out their tutorial. But we have our UMAP. This is when you would do your cell annotation to figure out what these clusters actually correspond to. These data have already been annotated. So we have the different cell types, and we know that these are the starting cell types in this differentiation trajectory. So for pseudotime, we're just going to pick this cell at the very top here as the root cell. So we're going to go ahead and initialize our pseudotime object, and we're using XUmap. If you do the force-directed graph, you would change this. And we're going to base it on laden. I could do this cell type annotation as well, but we'll just stick with laden for now. And these data, there's only one lineage. You know, it goes from this cell type to this cell type. Of course, depending on your data set, you could have multiple lineages. So for Pseudotime, we need to create a lineage dictionary, and I'm just calling this lineage the one. The name doesn't matter in this case, there's only one, but it's just going to be a dictionary of the lineage name and then the items that correspond to that lineage. In this case, the laden categories, because we specified laden up here. And I'm just putting a dictionary together using all the laden clusters because it's only one lineage. So if we look at that, it's just a one entry dictionary with all of the laden clusters. So we're going to set that and plot that. Of course, it's all just one lineage, so we only see one coloring here. And now we have to pick the root cell. We're going to pick this root cell. If you had multiple lineages, you would pick multiple root cells. So we just have to get the cell barcode for this cell right here. And there's multiple ways to do this. This one's actually a tiny bit more tricky than many cases. For example, if this was your starting cell population, you could pick the cell with the highest Y value. Or for example, here, we could pick the root cell based on the lowest Y value. And that would look something like this, where we take the UMAP coordinates. And in this case, this is the Y value. If you wanted the X value, you would change this to 0, but Y is 1. And then we're just getting the ID or the index of the cell with the lowest Y coordinate. But of course, that doesn't work for this graph, so we're not going to do that. We're instead going to take the cell with the highest X and Y sum, because this is X, this is Y. I know that this cell here will have the highest sum of X and Y. If we wanted to take the lowest sum, it would be somewhere over here, but the highest should be here. So we're going to take the X and Y sum and create that sum for every single cell. So we're going to get the index for that cell, which is just going to be an integer. So what position in the data frame is that cell? And then using that index, we can get it from the obs names, which is just a list of the cell names in our data set, and we'll get the actual cell name. So if we look at root cells, it's just for this lineage. This is the root cell barcode. Again, if you had multiple lineages, you would have multiple entries in this dictionary here. The two ways I showed here should cover roughly 95% of use cases probably, but the CellOracle documentation actually has an interactive plotly method where you can open up the plot in a window and actually click on the cell to get it. A little more complicated, I think, than just doing it this way, but theoretically easier, especially if you have a very complex graph, or maybe if your root cell is somewhere in the middle or something. But anyways, we have our root cell for our one lineage. So we're going to set that in our Pseudotime object, and then we can plot it just to make sure it picked the right one. So yep, it picked the right cell. And then we'll calculate the diffusion map for the A data of our Pseudotime object. And then we'll calculate the Pseudotime for each lineage, or just our single lineage. And then we can plot that. So we have our Pseudotime for our single lineage, and then we're going to get two graphs. You would get one additional graph for each lineage. We only have one lineage. And if you wanted the actual Pseudotime values, that's going to be under pt.adata in the obs data frame. So we have our Pseudotime values here. Okay, so this is, basic Pseudotime analysis. You could do a couple more things just based on Pseudotime, but we're just going to use this Pseudotime mostly for our downstream in silico knockout analysis. So if you have your own ATAC data, it can actually even be bulk ATAC data. But if you have ATAC data that correspond to your cell types, I highly recommend following their base GRN input data preparation. Again, you don't need to, but if you have the data, it will make these analyses a little more accurate. For this tutorial, I'm just going to use an already calculated GRN that they provide. So if you want to look at the GRNs available, one way you can do is just type this out, then hit tab, and you can see the different GRNs they have, which you can potentially use for this. My data are mouse data, so we're going to use this mouse SC ATAC Atlas base GRN, and we're just going to call it base GRN, and this is just going to be a data frame of ATAC peaks, corresponding genes, and interacting transcription factors. So we have the base GRN. This is what you need If you're making your own GRN from your ATAC data, this is what you should have at the end before we go any further. But let's go ahead and use this base GRN. You can make a new A data, but just for simplicity, I'm just going to change the X in our A data back to the raw counts. I wouldn't make a new object if you're planning to use the A data further anyways. Let's initialize our Oracle object, and then we'll import our A data object with the raw counts. This is important. You're supposed to have raw counts in the X slot. That's why we did this up here. And it loves to throw this error. I promise you these aren't log transformed. If we look at the max value, it's 2286. That's not a log transformed value. Now we can import our GRN. We're going to use the perform PCA function, and then we're going to select the PCs to use. And in the CellOracle documentation, I'm just taking this directly from that. They provide this function that automatically selects the number of PCs, in this case 26. And then we have to select how many clusters to use in our KNN, and they recommend taking how many cells you have in your data frame, in this case 3696, and multiplying that by 0.025. And so our auto-selected K is going to be 92. So we can use that in our KNN imputation. We already picked 26 for this and 92 for this. And then we're going to calculate the GRN for each individual cell grouping, in this case, again, based on latent. So this is going to be the longest part. It's probably going to take a few minutes. Depending on your data set, it could take a while. All right, so after that's done, we have to filter these GRNs down a little bit. And we're going to use the default values. So we're getting rid of any links with a p-value of greater than 0.001, and then keeping only the 10,000 top ones. And then let's plot the degree distributions. And here is each one for each of our latent clusters. And the degree is just how many edges each node has. So let's say you had 3 genes connected like this. This would have a degree of 2, this would be 1, and this would be 1. So there are some transcription factors or nodes in the GRN here that have 400 different connections. But you see the majority, or the proportion here, the majority have a much smaller K. So you can look at this for your different clusters. We're just going to go ahead and get the network scores. And if you wanted to look at the data instead of the graphs, now we have the network scores. So we have the different transcription factors and the different network statistics. I'm not going to go into depth here about this. I do recommend looking it up. But basically, the higher the degree, the more connections a node or transcription factor has, the more important it is for your network. We can plot the different values. So if we wanted to plot these data for cluster 3, we can look at the different degree centrality. So you can think of this as importance for cluster 3. And we can do some simple comparisons between the GRNs of different clusters. So if we wanted to compare cluster 3 versus cluster 4 based on their eigenvector centrality, GHRL is very important in cluster 3, but not so in cluster 4. And likewise for these other transcription factors to a different extent. And if we wanted to pick a specific transcription factor to look at, let's say we were interested in MAFB, we can see these different statistics for the different clusters. These would be a little nicer if they were ordered based on the developmental trajectory. I could do that, but basically MAFB is low, and then later in the differentiation it increases. So those are just some basic GRN analyses. But the main reason we calculated that was to do this perturbation analysis. So we're going to initialize that now. So we're going to add to the Oracle data from the links and fit the GRN for simulation. And we're going to continue to use MAFB for our example here. This is the imputed count, not the actual counts. It's important to use this. And for any perturbation, we can go from 0 to around double what the max expression is. So in this case, we wouldn't really want to go above 4, We could theoretically overexpress it, but I'm going to do in silico knockout instead. So we're going to take mathb. You could do multiple genes here as well. And we're setting the counts, or the imputed counts, to 0 for this shift simulation. And then we're going to estimate the transition probability and calculate the shifts and the embeddings. And for the plotting, again, I'm just taking this directly from the CellOracle documentation. So we have our knockout versus a randomized simulation vector, or you can think of this as a randomized control. And the thing we have to modify here is the scale. If I make it small, these get huge. So you have to play with this. The cell identity shift is very large over here, but not so large in these cells. And if you remember from above, these are the cells that are actually expressing mathb, so that makes sense. If you want to do grid, instead of plotting a point on each individual cell, we first have to get these different parameters for the grid. So we're going to create a 40 by 40 grid, and then we have to calculate this mass and then the mass threshold. If we had a really small value, we would have an arrow everywhere in this 40 by 40 grid. But we want to pick one that covers most or all of the cells without having too many outside of the cell space like you see here. So it looks like we would want to pick somewhere around 6.2 to 9.4. I'll just go with 6.2. So we pass 6.2 as our min mass. And then this is what we're going to be left with. And then we're going to plot what we just plotted up here, but on the grid instead, which I think looks a little nicer. So it's very easy to tell the differences based on mathb knockout versus the randomized control. There's a huge effect on cell identity over here, not so much in these earlier cell populations. And then finally, we're going to use the pseudotime we calculated earlier to calculate developmental flow. So we can compare developmental flow and the perturbation effect. So to our Oracle object, we're going to add in the observation column pseudotime, and it's going to be exactly the same order as the pseudotime from our pseudotime object. So we haven't filtered any additional cells or anything. And then we're going to make a new gradient object using the gradient calculator, passing our Oracle object and specifying that pseudotime column. And then we're doing the same thing we just did for that Oracle object. We're using the same number of grid, 40, the same mass threshold, which is 6.2. You could change this to true if you wanted to, but it's going to be exactly like it was above. And then we can look at our pseudotime plotted in the grid. So this is our pseudotime that we had earlier, and then this is our grid pseudotime. We can then calculate the gradient, and then we have to play around with the scale for our developmental data here. It might be a little hard to see, but if you can see the arrows and they're not too small, then the scale's probably fine. Again, smaller gives you huge arrows, bigger value gives you smaller arrows. We can blow this up here. So here's our developmental flow based on our pseudotime. And now we want to find how perturbation affects this. So we're going to initialize this dev object. We're going to pass to dev our gradient object as the reference data. And then our perturbation data, which was the mathb knockout, is in our Oracle object. So we're going to add that to dev. And then we're going to compare the two by calculating the inner product. On the CellOracle website, they have a good little cheat sheet here for what that actually means. So if the arrows are in the same direction, then the inner product is 1. If they're in opposite directions, then it's -1. If the knockout changes the developmental flow in the complete opposite direction, it's a -1. And various in-between values, that's just basically the difference of the direction of these arrows here. And we can show those product scores similarly like we did before, where the left is based on the perturbation and the right is a randomized. So here you have to change this VM parameter. If it's low, you'll see color on both. So you want to set it to a value that's high enough so you don't see much color at all on the right side. Again, -1 is the burgundy color, means that there is a big impact on the developmental flow. Green would mean that it supports that developmental flow. And finally, we can overlap that with the vector field as well to show the effect of the perturbation as these arrows as well. Their documentation has some more visualization examples. This was a very basic walkthrough. I do want to point out that the dataset I used for my CVelo might not be the ideal dataset to do this kind of perturbation analysis on it. For example, in the datasets they have as an example in CellOracle, this dataset has 2 different lineages. The knockout is extra powerful when you can determine which way in the split is controlled by the gene of interest. So to summarize everything, we selected a root cell, we calculated pseudotime, we calculated cluster-specific regulatory networks, we did some basic comparison and analysis of these regulatory networks, and we used CellOracle to do this in silico perturbation analysis, and we examined how the perturbation affected the developmental trajectory of these cells.